I hope my audio is clear. Yes, can someone confirm? Hello? Can you confirm, please, if my audio is fine?
Sir, we can hear you.
Yes, okay, thank you. Um, Sorry for the delay. I'm still having some network issues, so there will be a drag here and there. Kindly bear with it. So in today's class, we will discuss about site-directed mutagenesis. Before going in there, I would also like you to notice, you may have noticed that in the Google Classroom, I have posted 2 assignments. Okay. We have already finished 2 assignments. Now there are 2 more assignments, and then final assignment will be there. So the assignments 3 and 4 are common for everyone. Okay, I have given CAA question papers of last year as assignment 3 and 4. So you are supposed to answer it and then submit your video description, you describing the answers. You should have solved it first and then recorded a video that you have done. So it will improve your, not only your ability to solve, but also your ability to describe and express your answers clearly. More importantly, I would also encourage you all to discuss with each other. I don't have any problem you seeking help from each other. The point that I will be interested is I have to upload the rubrics. In the rubrics, it is about with the confidence with which you describe your answer clearly. That is what is going to count for marks. So you need to exude confidence. You should be clear and articulated in explaining your answers. That will be the objective of the assignment. Okay, let's get to the topic. So site-directed mutagenesis. We are saying site-directed mutagenesis, but typically we should talk about everything, right? To start with, why do we want to even modify a gene? At one point, I was describing about the significance of gene or rDNA technology. Why do we need this, right? So the nature has its own ability, or it has its way of shaping organisms, in which it meant by changing the genome or the genetic information. It doesn't change randomly. Changes happen. Those that are good changes for that location and condition will be selected. That's basically natural selection, and that's how evolution works. And if we were to we need more produce, we need more better produce. So in that case, we are supposed to intervene and change things as we want. uh, so then one second, what is, did I miss something? Okay, so then in that case, that is how genetic engineering or rDNA becomes powerful, because we are able to put them into the way we want more expression. We can change the way the promoter is, what kind of promoter it is. We want to know when it should be produced. We can add regulators to activate or repress the promoters. We want it to be produced in a different cell, then we can change, we can choose the type of promoter we want. Okay, so which gene should be expressed, when should it be expressed, how much should it be expressed? These are all the things that genetic engineering and recombinant DNA technology can do, and that's what is the key to biotechnology, right? If you remove rDNA or genetic engineering from biotechnology syllabus, it will be anything else but not biotechnology. It will be chemical technology or something else. The core or the essence of biotechnology is one's ability to modify or engineer things engineer genetic information, engineer at the level of genetic information so that the structure will be or a compound will be produced. Okay. And there is one more addition to that. That is our ability, what genetic engineering or rDNA, as I said, it's about ability to produce, when to produce, how to produce, and all those things. What to produce also. Can we now better it? That is the question here. Can we now increase the quality or increase the specificity or affinity, some property of the genetic information, so that the product that comes out, a protein or RNA or anything, that has properties of our desire, right? Our desire would be usually to enhance something so that it either becomes cheaper or increase in produce or reduces the burden on of something else. So basically, we should be able to modify the gene. Okay, so we several protocols are available, and I am going to discuss a few of them. We don't have to go through all. If you understood a few key ones, then you can do many things. Let's check. Let's see some of those. And before going, let's be clear about what is a mutation, right? A mutation is a change in the nucleic acid. But usually the change should translate into a change in the information. In the product of it. So you have a change. There is a you may have a point mutation, and then it produces a some protein. There should be some difference when it is expressed in the output of it. Output could be protein or RNA, right? In either of these cases, if there is a change in the nucleotide sequence, but there is no change in the protein, then it is not useful to us. Yes. So typically, I'm now talking mostly in terms of how it is useful to us. I'm not talking in what happens in nature. In nature, there is change in sequence. Some sequences that they might be influenced at the end. In many times, there is no influence on the protein or the gene product. Okay. So usually a nucleotide change in the sequence of the nucleotides, the bases that are there, and that should be inheritable. Okay. And site-directed mutagenesis is there are two types of mutagenesis that we can do artificially. One is random. These are two strategies. I don't know if if I have a place, but yeah, I'll just take it here itself. What is the difference between random and and site-directed mutagenesis? Both are two different approaches. Both are useful. There is nothing like one is very useful and the other one is not. Now, say for example, I will try to use some analogy here. Say there's a bush, right? And you don't know what is in there. Okay, there might be a duck or something or not. And assume you are the hunter with a gun. And what do you do with that? How do you know if something, there is nothing else? What you got is a bush, and there might be something in it or there might not be something in it, but that is the only hope you have. In another situation, you can assume that there is a tree, and on the branch of the tree you have a bird or something. Okay. Well, I hope there is nobody from PETA in this class. Okay. But just for concept's sake, think of it like this. In the bush, there is something, an expression called beating the bushes. What does that mean? That means it and do like this. And if there is something, it might come out. And then you might catch it and use it for your purpose. Right? You don't have a clear-cut idea of what is going to happen, but you're hoping that there is a there is a possibility that you have that it can happen, and that is your only chance. So beating the bushes is randomly you mutate. If you have a gene, for example, from here to here, this length, and you randomly change things in different locations, most of them will result in a faulty gene. Okay, they might produce the gene product will not be useful, but occasionally you might have a kind of improved properties in the gene product. That is like beating the bushes. Whereas in the second scenario, site-directed mutagenesis, it's not that it always works out. In either of the cases, the same thing. But you know there is a tree, there is a branch, and you know you have identified that there is a bird. You can see it clearly. And now you have to just target that bird directly to catch it or shoot it. And that is something like site-directed mutagenesis. You have identified everything. You have a lot of information without which you cannot identify the bird and many other things. If you have enough information that this is the particular amino acid that is in the active site of this particular enzyme, and maybe if I change it to something else, change the amino acid residue in that position, it is probable that it is possible that it might change in affinity or specificity and so on. And then you're taking a very focused approach, and that focused approach is possible if only you have all the information in and around it, right? If you do not have information, then about what is going to happen, the approach to take is usually the beating the bushes. It is the random mutagenesis. And this one, when you have enough information and you are focusedly, you want to change some particular amino acid, then you are actually doing the site-directed mutagenesis. Okay? There are, in random mutagenesis, maybe I will try to take up somewhere, but it's not in the syllabus per se, because nowadays we have so much information. In the olden days, there wasn't enough information about a thing, a gene, for example, or a genome. We didn't even know which part of it is the gene even. But now genome sequencing and a lot of accumulated knowledge also, we have so much information already. And we have technologies like the techniques like bioinformatics, which will allow us to predict well which is the amino acid or which is the base or codon that we have to change. Some luck in a way. I don't want to use the word luck, but let's just say. So there are multiple ways, and the simplest one is this way. I want to change this sequence. I hope you remember in one case where I was describing about vectors that will generate single strands, right? I told you, and I will tell you again, the importances or applications of them. They can be used as probes in southern blotting, northern blotting, western blotting. Oh, sorry, not western blotting, southern and northern. They can be used for mutagenesis, as in the case of what we are seeing here, site-directed mutagenesis. Random also can be done. There should be one more, and that is sequencing. Right? Please note down a question. You should be able to differentiate in the same line. I would want to say, what is the difference between phage mate? Phage, second, again, I'm having trouble. Okay, I'll just dictate it. You can write it down. Phage mate, P-H-A-G-M-I-D. P-H-A-G-E-M-I-D. Phage mate, phasmate, P-H-A-S-M-I-D. Okay, you already know about cosmate. Okay, cosmate, phasmate, phage mate. You should try to know what is the difference in terms. This is in light of vectors. Okay, let's stick to site-directed mutagenesis now. So if I had a single-strand template like this, like we can use M13 phage we have used in the previous, in the vectors we have discussed about M13 phages as well, which are important for generation of single-stranded DNA. So if we had a single-stranded DNA and then we use a primer, so my goal is to add a different base in this location. Okay, I want to change this G into C. That is kind of, it's a point mutation, it is a substitution, and it is transversion, right? I hope you remember those things from our review in the beginning of the course. And also, what the simplest is to take a primer, or you design an oligonucleotide such that it is complementary everywhere except in this location. In this location, you have inserted a G. Okay. Now, if this keeps on polymerizing, it will polymerize something like this and make a double-stranded DNA. If this plasmid underwent replication, what would happen then? One of it will have, I'm trying to draw this one as a template. So that is the G, and the other strand would have been the C like that. I'm now drawing the other one here. This the lower strand. In this case, I'm putting it inside. It was originally the sequence here is G, and the outside strand would have been C. Right? This is the native or the wild type. I'll just put it as wild type. And this is the mutant. And the outside should have been the C. But because we have a single strand template, we can use a primer which is having a mismatch in one location. That is the point of mutation. And there we have put in G. So we got a strand, we made a double strand of it, and then allowed replication to happen. And in that case, one will be wild type and the other one will be a mutant. Then you should be able to select this and use it for your purpose. Okay. Three things that we will discuss, and I think you know these stuff. This one is of my special interest, probably doped oligonucleotide. Not that we will discuss slowly. So here you have a plasmid, and you have the gene of interest. You already know a certain regions or the restriction sites are available. You can cut this off and put in a gene of interest in that location. So the rest of the gene probably is something. Rest of the gene could be similar to that of wild type, but in the middle you have removed a cassette and put in a new cassette in there. It is something like, if you know about exon shuffling in eukaryotic gene regulation, it's almost similar to that kind of things. So we are not randomly deleting anything. It is, in a way, I'm trying to show it this way. We are going to cut this part out. We will remove it and then put in a new one, right? New part in this, in here like this. Okay. Now, when this new gene is a recombinant gene is expressed, a different product is formed with this information included and the native or the wild type information deleted. That's a form of mutation. We're exchanging fragment of gene altogether. That's kind of cassette mutagenesis. You're trying to remove a certain bit of the gene, not just a small point or something. And this is what I would want to pay attention. And I like this one. Not much is useful. I don't think people need to do all these right now. We have site-directed mutagenesis, like in the ones we will describe a little later on. But this is ingenious in terms of thinking, and that is why we should pay attention to how these people have done. And there are some things that I don't understand. So what they have done is, I hope you remember how DNA is synthesized, right? So we will try to recollect that. So first they have added T, like in this case. Then they will add DCTP, so C is added. And after that, they will wash it out, activate the 3' end also, or the 5' end of G. And now DGTP is added, so G is added here. And A is added here. Like this, they have synthesized DNA until this place. Okay, so I will try to write that sequence if it is ATC. Okay, so there are several of the same sequences. Okay, I'm not writing the sequence until the end here. And now what they did when this position has come, they added DNTP. What will happen then? If you add DNTP, one may get a G, right? I will one may get T and one may get A. They added, why does that happen? Because they added all DNTPs. Then they wash it off and again add deactivated DNTPs. So this one may get C, T, A, G. Okay, different oligonucleotides in this position may get different bases. I hope you see that we are having mutagenesis. We're changing the mutation. We are changing the base in. We are having several combinations of it. It's straightforward. Sequence is determined. We are doing that. From then onwards, we are going to have, in this position, there are 4 possibilities. In this position, 4 possibilities. And in the next one, they add either G or C. If you add, so when it is time to add some base here, they will add DGTP and DCTP. Some of them may get G, C, G, or C like that. So in this case, there are 2 possibilities. And keep on doing it. I hope you can see the number of combinations of codons they're making at this location, right? One can probably generate a large number of codons here. Okay. So this is about the technical part of the design. Actually happens that, assume that some people have identified that there is an amino acid that is likely to be crucial for, say, identifying a substrate or accepting a donor, accepting an electron, or it's a donor of an electron, which are usually the important things that influence the activity of an enzyme. So if I have identified, and in that location, I want to try every amino acid combination. And if I want to do it you try, how many? I have to try 64 different, we will say 61 different codons. We don't need the 61 different codons, so we can try 20 different codons. How many times do I have to synthesize this? Then I have to synthesize 20 different DNA fragments like this. And I have to also synthesize the corresponding one. That will be another 20 primers or 20 oligos, and total of 40 oligos I have to synthesize. Imagine, again, remember this. The goal is simple. In this codon, I want to try all the 20 amino acids. Again, I also want to try all the 20 amino acids in this codon. If I try to make that, how many should I make the oligos if I want to do it straightforward? And then I also want to have 20 and 20. You can count that. So somewhere around more than 400 oligos you should make. And you also have to make the complementary of them. That will be another 400 something. People have designed something, or engineers, or I think I find it very intellectual way of doing stuff. They did only one reaction or two reactions in total, one for the top strand and a gene synthesis reaction, okay, and one for the lower strand. We'll see how that happens. So in this case, you have one reaction. I hope you understood this concept, right? When the codon in question that you want to try different codons, they manipulated what they have added here. They add all DNTP, so you can get these kinds of mixture of oligos. So the first synthesis of first strand is fine because this is the strand, and in this location it could be any of these. These ones are the combinations. I hope you can see that 44 into 4 into 2 for one codon space, and the second codon is also there, and different genes. Here I have drawn only 4 of the oligos, but it will be in thousands. So you have everything represented more than once. So you are getting a lot of oligos. But it's easy to make the first strand in this way. What to do with the second, the complementary strand, that will become difficult, right? Because whatever you're having here, we don't know what it is, and it should match with the complementary strand. So what they have done is they synthesize this, and here when it is time to add in this location, they added inosine. I hope you remember that it is one of the nitrogen bases, the precursors for purines, I guess. And they added inosine in this place. Inosine can base pair with all the bases, A, T, G, C, something like that. So irrespective of whatever nucleotide is here, or base is here, or in this location, inosine can base pair with it, right? So no problem at all. You synthesize this, you synthesize the lower strand in this locations where mutation is expected. You would put in inosine, and now you allow these two strands to anneal and then ligate with it. I hope you noticed why they have these overhangs like this. They made these overhangs on purpose because they want to put it into this place. They want to ligate this DNA into this location. I hope you saw a lot of things. You should now, I will try to repeat once again, just in case you're lost. Modification in two codons. In those codons, they want to try all the 20 amino acids and 20 amino acids. So it is a large number of diversity is required. If one were to go by the traditional approach at that time, they should have gone synthesized so many oligos, and they should have somewhere around 400 or 800 DNA synthesis reactions. Instead, these people have taken a novel approach where they have manipulated how they synthesize the reaction. And in the position where they want to try all the bases, they added the whole DNTPs. So in different oligos, we'd get a different base like this. Similarly, the second one also. For the third one, they want to add either G or C. So they added only DCTP or DGTP. So I hope you see the diversity of oligos that are forming just in one DNA synthesis reaction. Okay, I mean one oligosynthesis itself, they are generating a diverse, even more ingenious, because they have exploited the property of inosine to base pair with any of the bases. So in the locations where a random base could have been added, such as in this case, in that position they have included inosine like this. So once this is ligated, upon ligation and then transformation, okay, once it is transformed in this cell, inosine within DNA is a damage, right? DNA will the repair mechanisms will see inosine as a um, is a modified base, so they will remove the inosine and add whatever corresponding base is there. C was there, and the corresponding strand, the repair mechanisms will remove the inosine in this location and will add G like this, right? So you are getting, then you can of course choose which ones, which are the oligos or the which is the clone that is satisfying your needs. You would identify those and go ahead for characterization and other things. Okay, this is a nice one, less discussed, but very there is a lot of intelligence in there. That's why I wanted to bring it to you. Okay, next is next are primer extension. It's much more simple, like we have discussed in the previous. The first one I opened is the primer extension one. I'm trying to describe it again. Here is a M13 single-stranded phage. You have the DNA cloned into it. It is in single strand. So you can choose a oligonucleotide with the mutation. This is where you want to make the mutation. A should be mutated to C. Then you add DNA polymerase, DNTP, and T4 DNA ligase, and then these two strands are synthesized. Second strand is synthesized, and this is a mismatch, right? And if upon transformation, um, if the mutation is repaired, then it is a problem. But if it underwent replication, then you would have one is wild type and the other one is mutant, and then you take this and one should be able to characterize and work on it further. Okay, so this is a PCR kind of, it's not a chain reaction, it is just one polymerization reaction and then putting it into transforming it into E. coli. So that is called as primer extension method. There is one more that is dependent on inside directed mutagenesis. We will see it at there. But there are also multiple things one can do. This is you can do multiple point mutations like what we have seen previously. We can do the same thing, but you can add more number of mutations within the primer, okay, or oligo, where within the primer you add this part. This region is an extension or insertion that you want to put. Similarly, one can also do deletion, right? So because this is the primer, you wouldn't have added this part. You will have complementary to this and you will have complementary to this region. So when this replication happens, when this for this plasmid, you would get a, to tell me if you can see my can you see my screen? Can anyone please message? Voice, please. Can you see my screen? Um, Power is gone here. Can you see my screen?
Yes, sir.
Oops. Sorry. Um, so now we will describe about the PCR-based site-directed mutagenesis. I think I have, okay, 10 minutes. Maybe what I'll do is I'll stop here because anyway, I think I'll take this up in the next class. I have to, there are three methods that we will discuss, and I would want to just talk about any of the questions if you have in the middle. Yes, please. Any questions regarding assignments or anything basically, or what we have discussed? I will have a question for you. Yesterday I asked a question to you, right? Or did I? Okay, I will just use this. Maybe I will not discuss any. Okay, there is one question I will have. You may note it down. So in the doped oligonucleotide mutagenesis, where diverse oligos are generated using or manipulating they have added N and in the third codon or C. Why do you think that is? Okay, I want you to try to find out why do you think they added G and C. Why not N in the third position also? Anybody? Any ideas? Have you noticed? You must have noticed it, right? My network is bad. Won't move ahead, but try to find out the question, the answer for the question. Basis of the codon, they have added N and N, but in the third one, they added either only G or C. Go to Google, search in Google, about the title of the method is called doped oligonucleotide mutagenesis. Try to find out why. Try to find more details about it, why they have done,